All right, great. So I'm glad to tell you about some of the work that we've been doing recently in the lab. We've spent a lot of time this week talking about one kind of cellular mistakes, which is mutations, and what are their consequences, how do they come up, and what is their fate in populations, and so on. And in today's talk, I'm going to tell you about some work we've been doing on understanding another kind of cellular mistake, which is mistranslation, which is when cells make mistakes in terms of the proteins that they're producing, right? So there's no genetic mutation, but during the process of producing proteins, cells very, very often make mistakes, right? And so protein doesn't look like what the amino acid sequence should look like given the DNA sequence and the RNA sequence, right? And so let me give you a bit of background. In general, in cells, there are many ways of mistranslating. So for those of you who don't come from a biology background, just a quick recap. In general, you have a messenger RNA copy, which is a string of bases, which is made as using a DNA piece as a template, right? So it's your gene. That's going to be translated by the ribosome. There's a start site, there's a stop site. There are tRNA genes, which are adapter molecules carrying amino acids of different varieties. They're going to use this anticodon, which is a triplet set of bases here, to match with the codons, the triplet codons that you find on the mRNAs. And the ribosome is going to mediate this interaction, right? So the tRNAs come in bearing the right amino acid. The amino acids are going to be attached by bonds, and then the tRNAs go off, and in the end, you get some collection of protein molecules, which will then be folded and so on. So there are many different ways of mistranslating in this context. You might have changes in the start or the stop site, which tells the ribosome where a protein should begin and where it should end. And this can happen through various kinds of mutations in the DNA itself. You might have a change in the tRNA pool. So if instead of having lots of pink amino acids in this pool, you have fewer varieties of tRNAs which carry this pink amino acid, you might then end up generating more mistakes because the ribosome, ribosome is more likely to incorporate these other kinds of amino acids instead of the pink one, right? So then you may end up with a protein which has many more of these green amino acids instead of the pink and the blue that it was supposed to have. And finally, you may have mutations in the ribosome itself, which may make it more or less likely to make mistakes. And some of these mutations are very well characterized in some bacteria. And so this is another way that mistranslation can occur, right? Or you may have lower mistranslation levels. So here are the kinds of approaches that we have used and a lot of people have used to understand the evolutionary consequences and evolution of mistranslation in general. So a lot of work in my lab in the last few years has focused on trying to understand ways in which bacterial genomes seem to have evolved to minimize mistranslation, right? If we have less mistranslation, if we want to have lower error rates, what should we be doing? For example, we may have a nice match between which codons are being used and which tRNAs recognize these codons. You remember the genetic code that you saw in a lecture earlier. So if there's a nice match, then it's less likely that ribosomes may make mistakes. If you have modifying enzymes which allow a specific tRNA to recognize and bind to codons that it may not otherwise recognize, that can alter how many mistakes and how often you would make these mistakes. So we've tried to understand how these modifying enzymes have evolved in bacterial phylogeny. If you have different kinds of GC content shifts, so you have bacteria which span GC content from 15% to 75% GC, that's a very big shift in the kinds of codons that you would use, right? So this ends up generating selection to change tRNA pools as well. And so we've tried to understand these kinds of dynamics, again, in the bacterial phylogeny, and ask at the experimental level, if we change tRNA pools or change codons, what is the consequence for the other parts of the translation machinery? How does the machinery cope? And so can we understand how the system configures itself to perhaps minimize mistranslation rates? And finally, we're sort of compiling all of this information together to understand, in general, is there an impact of selection for fast or slow growth on various components of this machinery, right? And so all of this work is sort of from the perspective of saying, well, errors are kind of bad. You don't want errors, let's minimize them, right? On the other hand, there is some evidence suggesting that mistakes can sometimes be good. And you, of course, seen this in the context of beneficial mutations, right, in DNA. Clearly, if you don't have beneficial mutations once in a while, you're going to have a very hard time adapting, right, in general, any population. So the question is, are mistranslation mistakes that are that are occurring during mistranslation, are they beneficial? Under what contexts are they beneficial? What are the underlying mechanisms? Could there be a general mistranslation benefit? And what are their longer-term evolutionary consequences? And so this is work that was led by Lasya in my lab. Lasya is here. She's a postdoc who joined a few years ago. And she came in with this perspective that, well, I want to understand what are the evolutionary consequences of mistranslation. We know it occurs a lot, seems to evolve under some kind of selection, perhaps. What are the parameters that one is talking about? And so to address this, Lasya generated mistranslating E. coli in different ways, or manipulated mistranslation in different ways in E. coli. So this is the same kind of thing I told you before. Now I want to highlight a small distinction here. So there are specific kinds of tRNAs which carry FMET, which is formyl methionine, and that is the first amino acid that's going to get incorporated in most proteins, right? And so any methionine in the rest of the protein is going to be brought in by a different set of tRNAs. And these are called initiator tRNAs, and they're very important because initiation is where proteins start, right? So if you have problems with initiation, you're going to generate mistakes of various sorts when you translate. And so Lasya made a mutant in which she knocked out 3 of the 4 for FMET tRNA genes in E. coli. So that means that this E. coli cell now has much fewer sort of amounts of FMET in the cell and has problems with starting translation, and as a result also has problems with frame shifting and so on that she's figured out in the lab. So you have a mutant, what I'm going to call a mutant for the rest of this talk, which effectively has problems with mistranslation. It has much higher levels of mistranslation, right? So that's one way we can manipulate mistranslation to ask what is the evolutionary consequence. Sorry? It also reduces overall translation, yes. Okay, so here's another way we can sort of generate mistranslation. We can add analogs of amino acids. So we can add canavanine or norleucine. And effectively what these do is they kind of look like amino acids. They get incorporated in proteins, but clearly they're not going to be functional, right? Because they don't behave like, they don't allow the protein to function normally. And so we can increase mistranslation by adding these analogs in the culture. We can also change mistranslation levels by making mutations in the ribosome that make it more or less accurate, right? So we have hyperaccurate or hypoaccurate ribosomes, which are more or less likely to make, or less or more likely to make mistakes. Yeah. So these are the different ways in which we can now manipulate mistranslation, and then start asking, okay, now what happens, right? What does the system do? So what we find after some other kinds of screening, which I'm not going to go into, we find that mistranslation seems to increase survival under different kinds of stresses, and particularly under stresses which involve damage to the DNA. So let me walk you through these plots. I'm focusing here only on the wild type and the mutant as a comparison. Remember, the mutant has very low levels of initiator tRNA, high level of mistranslation. So here's percent survival in most cases on y-axis. So we're taking a culture. We know how many cells are there to start with, right? When they're not stressed, everybody's happy. And then we're stressing them either with UV radiation, which induces DNA damage, with hydrogen peroxide, which does something similar, and with ciprofloxacin, which is an antibiotic, which also generates double-strand DNA breaks, right? So effectively then we're causing cells to die, and this death is much higher in the wild-type cells, which have their normal tRNA, everything is fine, compared to the mutant, where on average many more cells survive. You see something similar with hydrogen peroxide and also with ciprofloxacin. Yeah?
So do your mutants grow slower?
They do grow a little bit slower in the absence of stress, yeah. So they pay a cost in LB, which is like nice media.
Do you think these effects could be caused by slower growing?
No, yeah, so good question, yeah. So when we reduce the growth rate of the wild type in a different medium, we don't see these effects. All right, any other questions? Everybody with me? All right, cool. So what's going on here, right? Why is the mutant doing better in general? And so we asked, well, what kinds of mutations are showing up? Because, for example, with ciprofloxacin, which is a very high dose that we're giving, most cells are dying if you look at the wild type, right? Survival is quite low. So what is known, very well known for ciprofloxacin resistance in literature is that you tend to have mutations in a specific gene, which tends to be the target of evolved antibiotic resistance, which makes cells resistant to ciprofloxacin. So simple question was, if we have colonies on an agar plate which are now ciprofloxacin resistant, that they're surviving, are they showing this mutation that we expect? And indeed they do, right? So I'm just pointing out this cartoon of a gyrase gene, and the wild type and the mutant both, all of these surviving colonies that we tested they all have mutations in the gyrase gene. Okay, so they all have a sort of a similar method through which they're becoming resistant. And from some early assays of rifampicin resistance, we see that the mutation rate is also similar in both the mutant and the wild type. So simple high mutation rate cannot explain this big difference in survival. So what is going on, right? Somehow they're surviving better even though they're getting the same mutation and the mutation rate is similar. So we tried to ask, is it early survival or is it later survival? Because what we're measuring here is after some 24 hours of growth on this ciprofloxacin, right? So when we measure early survival, we find that that is where this critical difference shows up. This is within a few hours of introducing ciprofloxacin in the cultures. So here is your wild type cell. I'm also showing you data now for these other strains, the other ways in which we manipulated mistranslation rates. Here's cannabinon. If you introduce more mistranslation by adding cannabinon or norleucin, you increase the survival rates for the wild type. On the other hand, if you make this hyperaccurate, the ribosome in the wild type, you drop these survival levels, right? Similarly, in the mutant, if you make the mutant ribosome hyperaccurate, where it's not mistranslating as much, overall, again, you see a drop in the survival, right? So this is just to show that it is mistranslation in general which has this effect. It's not something specific to the mutant which we're using. There are few mutations in the ribosome which are known to reduce the mistranslation rates in the ribosome, and others that increase it. This is something that other people have characterized for a long time. Yeah, so actually all of the ways you're manipulating mistranslation here do slightly different things in terms of mistranslation, right? So the ribosome, making ribosomes hyper or hypoaccurate is going to be a global effect, right? Where it's going to be sort of active throughout. When we are adding cannabinon or norleucine, this is only going to be relevant for the amino acids which they're mimicking. And so this should also be generally a global effect, but not the same as the misinitiator, like the initiation mutant, which is going to be largely affecting initiation. But it also frame shifts. So it also, we know from other work in the lab that that it frame shifts. And so, yes, there are different categories of mistranslation. And we don't exactly know because it's hard to measure using proteome data where the mistranslation is happening. So that's something we've struggled with for quite a while to sort of figure out, precisely what is each of these guys doing. Very hard to quantify. Any other questions? Okay, good. So then we're saying, okay, so this is early survival, right? And so here's what we're suggesting as the model of what is going on. So you start with populations of sort of equal numbers of cells of the wild type and then the mutant, which has high mistranslation. You start introducing them to stress. And what we're seeing is very early survival differences where there are no mutations yet. And we tested this looking at, again, sequencing some of these colonies at these early stages. Don't find any mutations here, but there is greater survival, as you see here, in the mutant very early on. And what we think is going on is there are these higher numbers of survivors as a result, which means that the population size is larger. And that allows the sampling, a better sampling of mutations, which are ultimately going to get fixed in the population and which allow the colony to grow on this agar plate. Right? So it's this very subtle effect, sort of very early during response to a stress, that is somehow allowing the mutant to have a higher frequency of sampling the same set of beneficial mutations. How does this happen? And so Lassia went on to figure out, well, what is the mechanism through which this kind of effect comes up? Here is what we know in a very sort of very coarse view of how DNA repair happens in cells. So remember, we're focusing on ciprofloxacin now, which damages DNA. So if we want to understand how resistance emerges, we need to understand, or how survival emerges, we need to understand what is happening at the molecular level. So here's DNA damage. You have a bunch of proteins that get activated, suppressed, all of that. Ultimately, it ends up starting this SOS response in the cells, right? So as the name suggests, it's a response to stress. This kind of response ends up activating DNA repair as well as a few other modules in the cell. And this we thought was maybe relevant for what's going on here, right? Because we're damaging DNA by adding ciprofloxacin. And so what Lassia has shown then is if we take wild type cells and we inactivate the SOS response by making mutations in the pathway, in the SOS pathway, you end up decreasing ciprofloxacin resistance. Again, if you suppress recombination, which is how this pathway works, so it's using recombination to repair the DNA damage that we're inducing, you end up suppressing survival. Same thing in the mutant. You have higher rates of ciprofloxacin resistance to begin with, but then you lose this resistance when you suppress the SOS or the recombination-based repair, right? So suggesting the SOS response is important. And the reason this is interesting is because the SOS previously was unknown to do anything to do with mistranslation, right? It was not known to be really responsive to mistranslation, and so this was kind of a surprise. And so what we think is happening then, and so in other work, which I'm not showing you data for here, last year showed that if you look at the levels of these different proteins or enzymes or components of this SOS repair pathway, if you measure how soon are they activated after exposure to ciprofloxacin, you find that the mutant is activating them earlier, okay, and activating them at a lower level of the stress. So a little bit of ciprofloxacin and the mutant cells are already near the threshold for SOS response, so they start the SOS response faster, and therefore we think they end up damaging the DNA repair, the DNA damage, repairing the DNA damage much faster than the wild type cells, right? And we think this is where this early survival, the small difference ends up showing up, and then that translates at a later level into a larger pool of cells which are able to mutate and sample these beneficial mutations. Good idea. Okay, so then here's a cartoon version of the summary that I just gave you. I don't know if some of you know this tale of Durvasa Rishi, who is, I don't know, in mythology, so he's like this, well, he's the guy who's always sort of angry, baseline angry, and anything shows up and he's like sharp, sharp. Sorry, I can't translate sharp for those who don't understand sharp. But he's basically, yeah, well, curses or whatever, right? And so here's the demon, and these are the peaceful guys. This is like your wild type, this is the mutant, and the mutant is sort of already primed to just throw curses, right? And if there's a demon that shows up, you know, he's gone. And these guys live better, and these guys, the calm ones are kind of dead. Anyway, so for more details, please catch Lasya or catch me during the coffee break. And there's also a bioRxiv preprint, which has a lot more of the gory molecular details if you're interested. And so then the next question was to say, well, what are the consequences of these, right? So fine, at a short-term level, there is a better stress response, right? The cells are able to repair DNA damage much faster. You can see that in the long term, maybe this has sort of major consequences. And I'd just like to remind you at this point that these are non-genetic changes that we're talking about, right? And this has a huge impact, at least on short-term survival. So another thing to highlight also here is that these are very random changes that are happening, right? So it's like a very stochastic process of mistranslation. So we don't know which proteins are getting mistranslated. We're pretty certain that different cells of the mutant all have different levels and different kinds of proteins which have been mistranslated, right? So it's not like a single mutation, a single genetic change, which has sort of a, in some sense, predictable effect on what's going to happen in the cell, right? If you have a mistranslating phenotype, you're going to have a very unpredictable and very wide range of impacts on the cell. And so it's interesting to think about what's going to happen in the longer term with these kinds of mistranslating cells. And so then the question is, well, does mistranslation alter the adaptive trajectory if it has these short-term survival consequences? And to test this, we took replicate populations of the wild type and the mutant, and we evolved them in this batch culture scenario in 48 well microplates, not flasks, but I couldn't find a nice picture of micro well plates, so whatever. So yeah, so we evolved them for a few days, right, and a few transfers, and we evolved them under different kinds of stresses. One, we have sort of as a control no selection, so they're evolving in nice LB media, which is rich, everybody's happy more or less, except remember that the mutant has a cost in terms of growth rate in LB. You have constant antibiotic selection pressure, so you have ciprofloxacin, ampicillin, or kanamycin, and there's only one such antibiotic which we're providing at a relatively low dose to each of these lineages. And then we have fluctuating selection, where we might expect that the impact of mistranslation might be more important because it's generating, you know, this huge statistical proteome, this diversity, which might be more relevant and more interesting when cells randomly cycle through or experience one of these stresses, right? So what we're doing here, just to clarify, is every transfer, we're using a random number generator to then say, okay, you know, are the cells going to get this antibiotic, this antibiotic, this antibiotic, or no antibiotic, right? And so they go through a series like this, where every time we We toss a coin effectively. And then we ask, well, what happens, right? And these are data that are still very new and raw, and we haven't sort of sorted through them very well, but I'm excited about them, so I wanted to share them with you. So here's what we see overall, right? So let me walk you through. There's growth rate as our simple measure of fitness on the y-axis. Here's our time course of the experiment. I'm just showing you data for beginning and end of the experiment. This is LB, which is no selection, and these are the 3 constant selection pressures, right, antibiotics. And I don't think I have to really describe what's happening. Is all of them are adapting to some extent where their growth rates are increasing. And the mutant and the wild type are maybe in some cases having slightly different trajectories, but both are basically increasing to different degrees in the different habitats. Now what's interesting is if we look at the genome sequences of some clones from these evolved populations, we want to ask which mutations have come up, which ones may have fixed in these populations or are floating around at high frequencies. What do we see, right? So in this case, in black is the mutation that we observe, or one of the sort of top mutations that we see at very high frequencies in these populations in the wild type, and the red shows you the same case for the mutant. So you see here that these 2 sets of mutations, don't worry about what they mean, but they're basically mutations in these genes that are showing up, right? So these 2 seem to show up at very high frequencies across the replicate populations, whereas the mutant seems to always have this 70 kb deletion, which includes a whole number of genes, right? In kanamycin and the 2 other antibiotics, we similarly see that, okay, there's some overlap in which genes are seemingly targets of selection. There's FUS-A, which is a known sort of antibiotic resistance gene for kanamycin, but again, the mutant seems to have this 70 kb deletion. In ampicillin, DNAJ is a protein that is targeted in both cases, but then there's some variation in other genes which are also acquiring mutations. And finally, going back to our old case of ciprofloxacin, you see again the gyrase mutations coming up in the mutant, fixing in every replicate, whereas, in the wild type, and the mutant, on the other hand, almost never samples gyrase A genes, right? It samples a different gyrase gene, but very often it also samples a completely different set of genes involved in efflux pump regulation, which is a MARA gene, right? So, and then finally in the fluctuating selection regime, this plot is a little bit different, so let me explain what's going on here. Remember that in the fluctuating regime, every generation or every transfer rather, they're seeing a different antibiotic, right? So for a few generations, they see, let's say, ampicillin or kanamycin here, then they get transferred to ampicillin, then they have only LB, then ampicillin again, and so on. And so at the end, we're still in the process of measuring the fitness of all of these different strains, evolved strains, on each of the 4 regimes that they experienced growing up. What I'm showing you here instead is the trajectory of growth rate in the sort of process of evolution, right? So as these populations evolved, you see this big jump in fitness increasing. Well, of course, part of this is simply the fact that kanamycin has a bigger sort of impact on growth rate and suppresses it much more compared to ampicillin. So you see this big jump, but then there's some degree of adaptation that perhaps is happening across this evolutionary time, right? And so again, at the end, if we sequence the mutants that have come up, we see different sets of mutations showing up in the wild type and in the mutant with some overlap, right? So what I think is going on is altering mistranslation levels, alters mutational trajectories, but not the adaptive trajectories too much, right? So if you look at the populations themselves, the growth rate dynamics, they look quite similar, right? Once you account for the ancestral growth rate, which is always lower in the mutant, the degree of change that has happened in the time frame we have is not very different between the wild type and the mutant. But the mutations that they are sampling seem to be, or fixing, seem to be a little bit different in some cases. And so the question is why? So one of the simple things well, there are 2 possibilities, right? The first possibility is, well, maybe the mutation spectrum is different. Because the mutant mistranslates so much, maybe it's going to change its DNA repair machinery, or its polymerases are not functional as normal. Maybe it's sampling mutations that are of a different kind, right? And maybe that's why the kinds of mutations you see getting fixed in the population also end up being different. So we try to test that using mutation accumulation lines. Do you guys remember what those are? I know they were mentioned in the lecture, but yeah. So basically, you're passing You're passaging bacteria through serial bottlenecks, right, of a single cell at every point. Continue this for about 2 months, and at the end, you have these 50 lineages which have all been bottlenecked for some time, and we sequence them and identify all the mutations that have happened, and we sort of create this spectrum, what's called a mutation spectrum, to say, well, what is the ratio of transitions, transversions, GC to AT mutations, and so on and so forth, right? And again, these are data that we received very recently, but a quick preliminary analysis suggests that we do not see major changes in the mutation rate as we expected or the spectrum, right? So it's unlikely that because of this sort of fundamental difference in which mutations are being sampled, you see these differences that we observe. So then the other alternative is, well, maybe it's a background effect, right? So because this is a mistranslating strain, the same set of mutations that are super beneficial in the wild type are no longer as beneficial in the mutant. And we don't understand why, but that's our current working hypothesis. And it's kind of cool because it suggests that, well, these different strains, when they're mistranslating, are perhaps occupying different parts of the fitness landscape or the mutational landscape, right? And are therefore sort of finding some local peak where they are and are not able to sort of jump to perhaps what's a global peak which the wild type occupies. All of this is conjecture, right? So we need to do more tests to figure this out and test it properly. But what we have so far is consistent with this idea that it's not because of which mutations are sampled, it's because of which mutations are fixed in the different populations. And that may be a function of whether you're mistranslating or not. Yeah. Okay. So that's where we stand. And we now need to sort of go back to these populations, look at the trajectory, when did these mutations show up? Did, for example, at a very early stage, did the mutant also sample similar mutations as the wild type, but just didn't end up fixing them? We need to make swaps where we take the mutations, these top mutations that come up in wild type and mutant, and sort of move them across the different backgrounds and ask whether this idea of being in different parts of the landscape, does that hold? And so all of that work is happening now. And yeah, and so I think this is really exciting, and I'm super excited to also hear your feedback on this and suggestions for what we could do next. All right, so what I hope I've shown you today is a new and general link between mistranslation and SOS response. And like I said, this is really exciting because it allows a sort of a plausible mechanism for the fitness benefit of global mistranslation under stress, right? So over the last few years, there has been quite a bit of work suggesting that under specific kinds of stresses, a specific kind of mistranslation of a specific protein can be beneficial, right? So let's say you have rifampicin, an antibiotic which targets polymerases, and if you have a specific kind of mistranslation in RNA polymerase, you're no longer susceptible to rifampicin, right? So a strain which particularly mistranslates this polymerase is going to have a good day, right? That's not something that can explain why or under what conditions mistranslation levels globally can be higher or lower, right? And so here's a mechanism, because SOS response is a very central bacterial pathway that responds to multiple stresses and which may be activated by mistranslation of any sort, right, beyond a certain threshold. We think this is a very nice way that perhaps because cells are experiencing stress very, very frequently, they may be able to use this existing pathway to then respond faster to stress. And perhaps, at least this makes it plausible that mistranslation rates being slightly higher could then be evolving under some degree of positive selection, right? With, of course, various parameters that are important, such as the frequency and the degree of stress. And finally, I've shown you that mistranslation can alter adaptive trajectories. And the extent of this and the generality of this, we're still exploring. One thing I will mention, I didn't show you data for, we also evolved some of the other mistranslating strains, right? So I showed you data just for the mutant and the wild type here. But we also evolved the hypoaccurate and the hyperaccurate strains, and they show also distinct sets of mutations that they seem to acquire, right? But they're distinct from the mutant which mistranslates. So probably a mutant is funny in its own funny way, but if you manipulate mistranslation levels in different directions, in different ways, we still seem to get different sets of mutations fixing compared to the wild type, right? So that also is an interesting result which we need to sort of explore further. I'll end by thanking all the people who've done this work. Lasya, all credit to her for really bringing in this exciting bit of set of ideas in my lab, and I'm super happy to have learned so much during this project. Parth is a project assistant who's helped Lasya do all of this work. Sharvari was a master's project from IISER Pune, project student from IISER Pune who came and did this phenomenal amount of experimental evolution in the lab in a short period of time. And Joshua, who some of you have met in this course before, is a PhD student in the lab who ran the mutation accumulation lines and is analyzing the data. We're also grateful to people from the next-gen sequencing facility at NCBS. These are all people who helped with strains and with advice and with different methods and techniques. And Anjana and Sunil, my colleagues at NCBS and INSTEM, who have been really nice and helpful in discussing this work. And so, yeah, they know way more about this than I do in terms of the cellular components of what's happening. So it's really fun. All the funding. And then finally, my lab group, which is a fun set of people to work with. And I'm grateful to all of them for just being there. Thanks a lot. I'll take questions. I think I have time. Yeah, maybe 5 minutes.